Hi, everybody. My name is Johnny Ullman. I am the Assistant Director for the Triangle Center for Evolutionary Medicine at Duke University. And you probably, hope, have noticed me at previous Club Ed Meds. I've been an organizer and along with many others. And it's wonderful to do this and have our group come to this every other week or so. This is fantastic. And keep in mind that we're going to be sending out the next semester schedule shortly I think hopefully over break sooner than later. So keep an eye out for that. But welcome again. Welcome to Club of Med. I'm simultaneously admitting people as I speak. So if I'm looking all over the place, that's why. Thank you again for coming to Club Ed Med Conversations. Our goal here is to connect evolutionary medicine community and share our exciting new research and awesome people that are part of our group. Again, we try to keep this as informal as possible with the emphasis on the discussion and presentation of different perspectives. So Dr. Jake Scott will give a little bit of a break, about 40 minutes in or so, hopefully, and then from there we can have some questions. And if you have burning ones in the chat or you want to raise a virtual hand, I can call out and we can kind of talk in between. I'll interrupt Jake. Overall, so Club of Med is organized by the International Society for Evolutionary Medicine and Public Health. It's a great time to become a member and or to renew your membership. And please support the society by submitting your articles to the journal EMPH, Evolutionary Medicine and Public Health. We are organized by 8 evolutionary medicine centers and programs. And I want to recognize our newest collaborators. We have 3 teams. It's awesome. The Brazilian Evolution and Health Study Group, located at the University of Sao Paulo, University of Kiel, the Kiel Evolutionary Center, Trans-Evo, and the Evolutionary Studies Group at Vanderbilt University. So keep an eye out for them and welcome them if you can. All right, so please participate in our conversation, post questions, like I said, or any perspectives that you might have in the chat box, or raise your virtual hand. And feel free to tweet about this using the hashtag #ClubEvMed. So this talk is part of the Translated Wins in Evolutionary Medicine, and I will paste some more information about and links about more of this shortly. So it is with my great honor and privilege to introduce Dr. Jake Scott. He's a radiation, well, I'll let him talk more. Sorry, I'll let you speak more on that. But Dr. Jake Scott is a veteran of the U.S. Navy submarine force, and turned academic physician scientist, which we didn't get to talk much about, but I think that is absolutely really cool, amazing. Jake's lab pursues research decomposing the complexity of cancer through mathematical models and the biological and clinical validation of these models. There's a lot behind Jake, and rather than getting into all the weeds, I'll let him take over from here. So with that, thanks so much, Jake, and looking forward to your talk.
Right on. Thank you so much. And it's nice to see some familiar faces. I've got the whole gallery pulled up. It's nice to see some familiar faces and lots of names that I don't know, and I'm excited to get to know folks. So I'm eager to be part of this community, and I'm humbled to be here. So I'm gonna share my screen. How do I do that? Like this? You'd think it makes it easier. Oops. And I'm gonna ask, does everybody see what I'm looking at? Is this good, Johnny?
Yeah.
Are we looking?
Yep. That looks. I have your. There you go. That's better.
Okay, it's the full screen. You're not looking at the slide deck side. Okay, cool. Yep. So my name's Jake. I'll give a little bit of some background. The talk's going to be kind of organized into 3 bits. The beginning is sort of a little bit of philosophy about why I think evolution is the only thing worth studying in medicine, and then a little bit of background about me and what we study in our group broadly, and then we're going to go into a specific use case, which is evolutionary game theory. It's not the only type of evolution we think about or the type of mathematical model we use, but in this setting, I think we'll stick with one. And then I'm going to pose maybe an interesting question and link to a preprint at the end that you might find interesting, and have plenty of time for questions. Everything I'll present is shareable, open. I'm eager to be interrupted, and I'm excited to share some of our lab's work with you. So we're going to talk about this concept that I think really, I want to move a little bit beyond. I think the study of evolution is important and predicting evolution is important, but I'd love it for us to get one step even further and move toward evolutionary control. And if folks didn't see, Brandon Ogbunu wrote a really beautiful article in Quanta about 2 weeks ago. Actually, Johnny, if you could find that and drop it in the chat for people. There's a really nice article that highlighted some of our work, which was was pretty cool. But really, this idea that evolution is a stochastic, complex process that I think that if we think about it in the right way, might have some opportunities for, if not control, at least thoughtful and intentional biasing of a process, which might be able to help us in medicine in ways that that are meaningful. Okay, so here we go. Share whatever you want, interrupt whenever you want, reach out to me about whatever you like, and let's talk about evolution. So I think we're going to set the stage by really talking about a couple different things. We have cancer, which is at one point has been the #1 cause of death in America. There was a little blip back and forth recently with a disease people might have heard something about, but really it goes between 1, 2, and 3 every year in the developed world as causes of what I call maybe preventable death. And then further in the less developed world, we're reaching a huge crisis point with the rise. I know that not everyone on the call loves the term superbug, but we're reaching a crisis point when it comes to antibiotic resistance across species. And those are both the sort of motivations for all of what I do in our laboratory, or what we do in our laboratory. But it's also worth mentioning that, you know, the vast majority of infections are cured pretty easily with a couple doses of easy to get antibiotics, or just tincture of time and waiting for the immune system to do its job, which is a beautiful co-evolutionary system. And then further, you know, the vast majority of early stage tumors are cured with either a knife or radiation or some types of leukemias with chemotherapies. And so while the talk is about doom and gloom about how we fail, it is important to realize that we have a lot of successes in modern medicine. So as much as I'll say things like, you know, everything's terrible and we need to study it, we do a pretty good job. And I think this is an advertisement for what I do. This is an example of a boy I treated as a radiation oncologist. This is a kind of cancer called a Ewing sarcoma, which showed up about the size of his heart, right next to his heart. And we were able to, in 4 days, with no side effects, completely ablate this tumor, and it never came back in that spot. However, it spread throughout the rest of his body and took his life when he was 21 years old. And the reason it did that is because once tumors become metastatic and they've seen therapy, they fail. The drugs fail, and the drugs fail because the tumors are evolving. And so here's a picture that many people have probably seen of a quote unquote lucky gentleman who has metastatic melanoma, which is a kind of skin cancer which is spread all over his body. It happens to be in the skin and easy to see here. And I called him lucky because he had a specific mutation in a specific gene called BRAF, for which we have one of our amazing targeted therapies. So after a few weeks of pills, his tumors went away. He felt great. However, like every patient who takes these medicines in these cases, it came back and was no longer susceptible to that therapy. And this doesn't just happen in the case of targeted therapies in cancer. It really happens across the kingdoms of life, microbes, parasites, viruses, and even animalia and insecta, and these different kingdoms that also are, I would argue, maladies to us as humans. And I think that we're going to talk a little bit in this talk about some beautiful work done by Bob Gatenby and others, thinking about this idea of maybe it's our treatment paradigms themselves and the way we think about eradicating these pests and tumors and bacteria, where it's sort of a fiat that this will happen, and maybe rethinking that in some way. I also want to highlight a beautiful paper by a friend of mine, Mike Boehm. People are probably familiar with this as well, this megaplate concept. And at the bottom is just a simulation of something similar. One of my students coded up pretty quickly. You know, and in this example, you have a petri dish that's like 6 feet long, and you had some bacteria in there, and they basically were given the opportunity to move and live in some swim agar and fill this plate in. And they moved from the outside in this plate. It's not the best picture, somehow the background didn't come in right. But what you see is that on the edges of the plate, the left-hand and right-hand side at 0, these bugs were exposed to just happy media and swimming. And then they were able to just spread. And they can spread toward the middle of this plate, and they can move up a gradient of drug. And they went from 3 to 30 to 300, and all the way to 3,000 times the concentration of drug that would kill them normally. And they did this in the course of like a week. And if you think about how far 6 feet is, if you're a bacterium, Not only did these things get 3,000 times stronger than they ever should have been in a week, but they also moved like the equivalent of us going to the moon. And so this was a really trivial problem for these bugs, and this is one of our standard antibiotics. And I just want to sort of also show what they showed in this paper was that these bugs didn't solve the problem in one way. They solved it in hundreds or probably thousands of ways in this example. And I think once I saw this result, it really just showed me that the entire paradigm of personalized medicine and cancer is probably fundamentally flawed. Sorry, I will argue that it's fundamentally flawed in that these are easy solutions for large collections of unicellular creatures to beat. Evolution is way better than we are at these problems. And so I think instead of really thinking about and focusing on developing these new drugs, we should really focus on studying the process of evolution itself. And I think this group likely agrees. And I'll give you some ideas as to why, and further how I think how the study of evolution can inform many different stages of these diseases. And so I am a physician scientist, so I treat patients with radiation therapy, which is a local therapy. It does, like I said, it does a great job of eradicating what I'm aiming at, but it really does a terrible job, or it doesn't even attempt to address the problem of disseminated disease. And I want to sort of talk about the cognitive dissonance I feel between being a scientist and being a physician just for a second. And I'm going to use this cartoon for the next 3 or 4 slides, so I'll explain it. People have probably seen Mueller plots before. So on the left-hand side of this cartoon is basically a Mueller plot. And on the right-hand side is sort of a Frankenstein version of something they call a Surkos plot. And what I'm trying to describe with this picture is the idea of the increasing heterogeneity over time from the left to the right of the Mueller plot, starting with a single cancer cell. Speciation events occur, if you will allow me to use that word, during the course of this diversification and heterogeneity. And then further, once these diversifications occur, we now have heterogeneous mixtures of things that begin to interact. So not only do we have growth and fitness, but we also have an ecology that emerges from this from mutation and from selection. And, really this process takes months and years, weeks and months to get where we are when we're treating folks. But at the moment I see a patient, when they come to my office, we'll biopsy the tumor and at that moment of max diversity, because as soon as we start treating them, we're going to start collapsing the diversity in some way. But we really think about this moment. Now, I think personally that the left-hand side of this plot is beautiful. This is evolution in action right in front of us every time, a brand new scenario. Patients, however, really don't care. They want to know what we're going to do about the problem right now. And as a physician, I don't care. As a physician, I just want to know, okay, here's a tumor I have sitting in front of me. What do I do with it? How does this incredible diversity map to a phenotype that I care about? And in this case, that phenotype that I care about is therapeutic effectiveness. And so I'm going to talk over the course of the next 5 or 10 minutes about each of these different timescales, months, years, weeks, months, and then now, and then how evolution can inform each of those things. And I think my background is helpful to understand why we're coming at it like this. And so this is a nice XKCD cartoon that I've stolen. And I really started my life as a physicist in college. I was at the U.S. Naval Academy, and I was studying actually space physics and these very long length scales, and then went from there to a submarine as an engineer, and really started using my knowledge of physics to make war, essentially. And it really gave me pause. When you're 22 and 23 years old, your brain's probably not fully developed, probably still not fully developed, but at the time it made sense. But I started really thinking, is this the best way to be spending what I have? And so during those years as an engineer, I learned a lot, and I learned that how we can apply physics to these amazing systems. But I started to wonder if there was a way to better help humanity. And so after the Navy, I went back to medical school. I was about 28 when I started. A medic is essentially an applied physiologist, if you will, and an engineer of this biological system that we're all trapped inside. And then after medical school, I studied radiation therapy, but I realized that I wasn't really using my background in enough of a way to make sense. So I dropped out of residency for a while to study applied mathematics and mathematical biology. And now that's really sort of where my head is as a physicist who went through medicine, then did applied math, and then found the glory of evolution, if you will. And now our group does a whole bunch of different stuff. I'm, the ADD phenotype I have, and it's only satisfied by the application in lots of different ways. And so we use lots of different mathematical tools and lots of different experimental tools and clinical tools to come up with hopefully answers to the questions that plague our patients. And from my perspective, each of these tools can be focused to help study evolution. And we'll talk about a lot of that. Okay, so back to this set of timescales. So I think cancers and infections from a theoretical perspective are incredibly similar. They're complex adaptive systems. They change over different timescales. They grow in population. They interact with one another. And I would argue, and I will argue, that each of these different timescales over which they act are mathematically tractable in different ways and experimentally tractable in different ways. And so today we're going to focus on number 2 there, which is the harnessing ecology. But I want to give everyone a few glimpses of what we do in the lab at a more broad scale. So I'm going to zoom in on each of these contributions that we play with and each of these time scales that we play with just as a snippet, and also to highlight some of the folks doing cool stuff in the group. So at what I call the longest time scale, and I'm gonna use the term evolutionary time scale just to make it easier to refer to things. We think really hard about, from a mathematical perspective, these ideas of fitness landscapes or fitness seascapes, which are just temporally or functional representations of landscapes over dose or environmental change, and really think about how, if we could put our hand on the scale of those fitness seascapes, could we bias selection in the long term and sort of choose the winners of an evolutionary process. And so you see some of the great students. There's Rowan at the top, who's a postdoc with us, thinking about how ecology and evolution plays in this time scale. Yishan, who's on the call, is an MD PhD student, thinking really hard about how to move some of these models into more realistic pharmacokinetic models to really make the application to medicine more feasible, because some of these things are just a little too abstract. And then there's a little hat-tipped some of our work with the physicists here in Cleveland, really helping us use tools from quantum information theory and statistical mechanics to think about these problems in a meaningful way. And there's some recent preprints that people can see if they like as well. And in the intermediate, or sometimes I call it ecological or clinical timescales, which is going to be the focus of the majority of the talk scientifically, we're thinking really hard about how interspecies interactions can shape or maintain or collapse the heterogeneity that is present in these tumors. So one of the big problems is that these tumors are heterogeneous, and therefore maybe the red cell doesn't respond to the same drug as the blue cell, and so we end up with a problem where each of our therapies only does a partial job. But what we're finding is that the ecology itself and the interactions between the players themselves is something that might be A, targetable, and B, meaningfully used and understood in a way to help us control tumors. And so this is being worked on by a number of folks in our lab. That's Jeff Maltus, who's on the job market and is awesome. If anyone is looking for a very talented biophysicist who does bacterial and cancer evolution in the lab and in theory, maybe some of you have his application on your desk now. If you do, consider it. He's really great. And he's written a bunch of these papers. There's Artem in the middle, who's done a bunch of the work that you'll see. He's a lecturer in Utrecht now and a game theorist. And then Nathan Frokin is one of the ones who got away. He decided to be a surgeon. And then Nina Din, who's a PhD candidate who's amazing and is working with us on a lot of these problems as well. So a lot of these, the work you'll see has been done by them, not me. But I think that this intermediate timescale, again, where we'll focus is, I think, the most interesting is this ecological perspective, and we're going to come back to that. And then the final time scale is the now time scale. And the question is, as a physician, this is what I care about the most. How can I take information about the tumor that's sitting in front of me, and I don't have any more months or weeks to play with evolution, but how can evolution help me think about therapy effectiveness? And we think about this in a bit more of a pragmatic way, but I still believe it's really convergent evolution that is guiding our thinking. And that is to say that there's many, many different ways that one tumor can be sensitive or resistant to a given therapy. And that lens of convergent evolution helps us really think about that. And in this sense, we're really deriving gene signatures and clinical decision support tools to help us take something that's super complex and heterogeneous and give us a sort of an easier answer, if you will, about what to do. What's the right therapy to give today, or the most effective therapy to give today. Okay, so that's a whistle-stop tour of the entire lab's work, or the majority of it. And I want to bring us back now to this clinical or ecological timescale, where we're going to focus the next 20 minutes or so and talk about some of the specific science and then stop. But I wanted just to give a second, if anyone has any questions or thoughts they wanted to share about the intro, which is now done, I'd happily take comments, or we can just push on to some of the science and then do it at the end, whatever people want. I'll give an uncomfortable amount of silence so someone else can talk.
I was just going to suggest that in your figure, you don't indicate consideration of non-tumor cells, but then I see in the lower right corner you do.
Yes, absolutely. We're going to talk a lot about how to get further along the lines, but some of the simple models we'll discuss first are tumor only, but you're right for sure, Neil.
But I mean, particularly with solid tumors. I'm not sure about, lymphomas and leukemias, but certainly for solid tumors. In fact, I just heard a talk in which the investigator was talking about the effects of collagen, covalent modification of collagen on tumor behavior.
So I didn't actually pay Neil to ask that question because the next slide really kind of comes to that, but it's a good intro to where we're going. And sorry, I didn't mean to cut you off, Neil. I just wanted to put this picture up because it's more fun to look at than the last one while you're asking.
That's fine. Thanks.
And his point's super well taken. And I think really this is the motivation for all of our thinking was, even when I was an early resident in 2008-9, we really did as clinicians think about tumors as just balls of mutated cells that were all the same. They were just a kind of cell that overgrew. And that's, honestly, I'd say still a fair number of clinicians still think of it that way, because during their training, that's how it was imagined. And in the last decade and a half, this idea of the tumor as an ecosystem has come to the fore. I think a lot of the folks maybe just discuss it. They don't use the word ecosystem as much as this community might, but certainly in oncology, we talk about tumor microenvironments and a number of other ways to think through this concept. And tumors are complicated things. Complex infections are also complicated things. I mean, you look at the beautiful work from Alvaro Sanchez and some of the other folks in that community looking at, complex infections. It's usually, if not more than one bug, at least quasi-species of individual bugs that all probably interact with one another in certain ways and also interact maybe in biofilms or in other ways that are really still ecological scenarios, maybe less so, but still. And so the bad news is that tumors are complicated. The good news is that the theoretical ecologists have been dealing with complex situations like this for years, decades. And so my job in life is to cheat off of the theoretical ecologists. No, sorry, stand on the shoulder of the theoretical ecologists in order to move science forward. And I think, I want to give some hat tips to some of the original work here from mentors of mine, Gatenby and the late Tom Vincent. This is really one of the first, or if not the very first paper, thinking about applications of these theoretical tools to tumors. And so this is an older paper from quite some time ago now, thinking about how tumor and normal cells might interact in some sort of luck of Volterra or replicator-like fashion. And on the right is maybe some pictures people have seen before, where these very simple models can really help us think meaningfully about complex situations. So the last slide I showed, you have 50 things or 100 things or 1 million things in this tumor microenvironment or ecosystem. But at some point, you don't need to know everything in order to have some meaningful control. So think about where these wolves and moose live. I see a marsupial there, so there's probably actually no wolves or moose there. But imagine you were in a mammalian-filled environment with wolves and moose. There's still also all these other species running around. But it turns out that if you apply some of these tools, you can still get a modicum of control over the species of interest without perfect information about everything else. And so I think this is a really important thing just to think about broadly. What can we measure? What can we control? What do we need to control? And I'm going to show you some examples scientifically in the next few minutes of a very minimal set of things that we might be able to control. But I would argue that, we don't need to know everything. You don't need to know exactly how many members of a grass species exist that the moose are eating for us to get some control over these two competing populations. But obviously more information is better, but there's a trade-off. Okay, so I want to just talk about a couple of mathematical treatments, and I don't want to go into the detail of these papers, but I want to give you some ideas of how we started thinking about this. So notice the date. This is 2011. That's Sandy Anderson at the bottom, who's my PhD advisor, or one of my two PhD advisors, and David Basanta, who is a game theorist at Moffitt Cancer Center. And I like to think about this paper because this is my gateway drug paper. So this is the first math bio paper I was ever on, and you can see I'm sort of solidly second there. And this is when I was still a radiation oncology resident, and I didn't really understand everything. Please. Did I hear a question? Oh, no. Okay.
Yeah, it might be feedback.
Okay, I'll keep going. But I want to focus on, it's not so much the model, but instead the question. So this is a game theoretic model. This happened to be a discrete time replicator dynamic where we could really think about how three different species, S, D, and I, competed and interacted. And so here's an example of what some of the temporal dynamics of this model might look like in certain scenarios. And we can study different things. And so in this model, what you're seeing is temporal traces of the three types, S, D, and I. Don't even worry about what they are. But the important thing is that what we could study is when you intervene in a model changes the overall steady state outcomes in a meaningful way. This is not surprising to a game theorist who's been working on these deterministic models, but to those of us in theoretical ecology, and particularly in oncology, it was meaningful that we could realize studying when the intervention occurs could be something that we have access to. And this happens all the time. Patients come in, they get the therapy, but we don't control when they're diagnosed. So we don't even know where along their life history this has occurred. And further in the same paper, we played with the idea of the duration of a given therapy and really showed for the first time that how long you give a therapy, so the width of that blue bar, could also drastically affect steady state dynamics. Basically what's happening to the mathematicians in the audience is you basically have a simplex and there's a couple different attractor states and you're kind of pushing things around, but you're blinded to the overall attractor states, but you're flipping them from one basin to another, if you will. So the questions you can now start to ask are sort of when to treat and how long to treat. And somewhat trivially, these answers depend entirely on what's in the heart of the model in this payoff matrix. So if you change the matrix around, the answers change. And so from some perspective, this is an obvious set of conclusions, but for us to be able to study these different things in a meaningful way was important. Moving on to a separate paper, this is a PhD student of mine called Artem, who I introduced earlier. Similar idea, slightly different model type, but same concept model-wise, but three types, and asking the question, if you have more than one drug, how does the order in which you give them change therapy? Again, it matters, but it also depends entirely on what you've written down for your model choice. Now, the problem with this model choice is that it's somewhat arbitrary. So the question that one could then ask is, does any of this really matter? Are we just playing with maths, which, by the way, is a fun thing to do and useful, but does this matter clinically? And I think that a paper came out a few years ago from Joel Brown, Robert Gattenby, and Jessica and Jin Song in Nature Comms, and I don't want to argue about the specifics of the paper because we could spend a few of these lecture series talking about it in particular, but I think the point here is that the idea was that a clinical trial was developed based on some of the ideas from models like I've described that showed, and the outcome, there's no argument about the outcome, which was that the men who were on this clinical trial for prostate cancer lived longer and received less medicine than the men who were treated with the standard of care, being maximum tolerated dose over time. And so the idea here was that they were playing with some sort of competition dynamic, which was essentially competitive release, which is the idea being that the sensitive or green cells on that plot were themselves out competing red. But when you kill them off with drug, they could then release the red to grow out. But again, if you remember, so I would argue that this clinical trial suggests that these models do matter and are helpful for us to think through how this works clinically. But it also, again, matters entirely on what you've written down as your overall model. And this is when I started my independent career, and we asked the question if we could really design a specific assay to measure the heart of those models? Could we figure out a way to go from the dish and empirically measure what goes into those models rather than invent them? And so this is some work done. Again, there's Artem, you've seen him a few times. Next to him is Jeff Peacock, who's now a radiation oncologist in Alabama. There's David again, and then my friend Andre Marusek at Moffitt Cancer Center. And this is one of the first works we did in my beginning of my independent career. But what we're going to do is ask the question of whether we can measure in that payoff matrix or not. And we're going to do so with a very, very simple system comprised of two types of cells, sensitive cells and resistant cells, and they started from the same stock. So there's the specific, for those who are interested, it's a lung cancer cell line that has a specific mutation, and that mutation is targetable, just like that gentleman I showed you with the melanoma. And what we did was we basically evolved resistance over 4 to 6 months. We didn't have much hand in there. That was just good old evolution doing our job for us. And then we're going to ask the question how these things play with one another in a dish, and in particular, we're going to ask a simple question about whether or not adding some normal cells can change that as well. To Neil's point, so to do that, I'm just going to give a simple explanation. So that what you see on the upper left in that matrix is the simplest, or a very, very simple representation of one of these competition payoff matrices. And so this is a representation. If you have those A, B, C, and Ds, you can write down ODEs that describe the population dynamics over time. So you have the D by dt of NP, which is the number of parental, and NR, the number of resistant. And so in the simplest sense, you have two ODEs, and really you can, because of some of the mathematical tricks that we use, assuming constant population size, you can rip that down to a single equation if you like. But at the end of the day, what we're trying to do is parameterize those two ODEs on the very top with some experiments. And so to do so, what we're going to have to do is do some co-culture. And so I'm just going to show you a little video. If you take red cells, which are resistant, and green cells, which are sensitive, and you put them in a bunch of drug, on the bottom you see this is a rather large dose of the drug. You see that the green ones start to slowly die and the red ones start to grow out. This is sort of what goes up must come down phenomenon. It's not helpful. It makes sense, but it's not helpful because we need to quantify this. And so in order to quantify this, we're going to count the reds and count the greens, as simple as you can be. We're going to do that with an algorithm so that we save our eyes. And I'm going to start building up this experiment over the course of a few slides. So if there's confusion, please stop me. So what we're going to do is we're going to count the cells over time. So the x-axis here is going to be time. The y-axis is log of cell number, or it's actually fluorescent area, but it's a good surrogate for cell number. And this happens to be in a scenario where there is drug. So yes, there's alectinib, and there's no CAFs, or cancer-associated fibroblasts. So you can almost think about, we're going to have basically 4 experimental conditions. Yes, no drug, yes, no fibroblasts. So there'll be 4 possibilities. And you can kind of think about the fibroblasts being like summer and winter, if you're a theoretical ecologist, different seasons, and the drug on and off similarly. So here's just how we're going to do these experiments. So again, this is no fibroblasts, lots of drug. You see that the green, and I apologize, sometimes the red and the green mean different stuff, but I'll try to label it well. It's because, one, colorblind friendly, this is not good, but the molecular biology is just the fact that we used green and red fluorescent proteins. And further, we always switched the markers to make sure it wasn't the marker itself causing issues. So in this case, the green is resistant and the red is parental. And what you see over the course of several hundred or 100 or so hours is you see a really nice linear fit on a semi-log plot, and showing the resistant with a positive growth rate and the parental with a negative one. Makes sense, lots of drug, we evolved them, we set them up like this. So now if we think about where to put these two, so we have a positive resistant growth rate and a negative parental growth rate, we can plot that on a single axis. And so now on the y-axis, we now have resistant growth rate. So we're seeing that we're plotting this in the positive above 0, and we have a negative on the x-axis parental growth rate. So it's to the left. So you see we're in the upper left quadrant of this plot for this specific experiment. If we do a different experiment and we take away the drug and we add back in the healthy tissue, we get a totally different answer. Everyone's growing quite happily. And now if we do lots of experiments, and I'll explain the difference between these squares, we see a reliable and sensible set of results. In every case, you can see those convex hulls drawn. In every case where there is drug on, we have a negative parental growth rate and a positive resistant growth rate. In every case where there's no drug, there is a positive growth rate for both types. And then you have this interesting case where it does span it, where you have healthy tissues and the drug. And we actually see this is sort of a well-known phenomenon that healthy cells, the fibroblasts, can actually rescue some of the drug effect. This is well known. This is not something we discovered here. But what's important is the difference in the colors of the squares. So you see there's dark squares and light squares. So we're going to look at just that alectinib one. So imagine that one on the far left. Each of those little squares, the opacity tells you the initial proportion of parental cells that were plated. Because we're thinking about ecology, we're thinking about proportions. We're interested to see if the fitness, the intrinsic fitness of a type changes based on who else is around. And what we find is that it does. And so each of those opacities, each of those squares is actually a pair of points here. So for example, the super dark one on the top is 99% of parental and 1% resistant. So it's the squares on the far right and so forth. And what you can do is you can actually now think about this plot and start parameterizing this equation based on the intercepts. And so now what we're able to do is go to these plots and pull off the entries for that payoff matrix directly from these co-culture experiments. And so this is very exciting to us because now we feel like for the first time, this is a method by which we can really pull these parameters out. And we make a lot of mathematical assumptions that we can complain about and talk about all we'd like, about linearities and first order systems and other things. But I do think that at least we have some sense. And I want to show you now of the 4 experimental conditions we came up with, some overall ideas that come out of this that I think are helpful when it comes to the evolutionary scenario. One, if we plot these now in a game space, we see that each quadrant represents a qualitatively different mechanism. So in the upper right, we have a stable coexistence. And interestingly, you see that the experimental condition that's there is no drug plus healthy cells. So that's a patient before they receive treatment, if you think about it. And that makes sense because before treatment, you do have some existence. You have a small amount of resistant cells coexisting and pre-existing in many cases. But in every scenario where there's treatment, we end up in the lower right corner where the parental are being outcompeted by resistant. And that's, as we know, what happens to our patients as we begin to treat them. But some questions that come out of this is that, can we really think about these therapies as changes to the evolutionary dynamic? Can we reframe our thinking about these therapeutics as evolutionary modifiers rather than things that just kill this type of cell or that type of cell? And can we think of that? Can we think of them in a new way and take advantage of that to be able to push evolution itself around? So those are some questions I want to leave for the group. And now the next few slides before I'm done are going to go a little quicker. Same exact experimental setup, but I want to generalize things somewhat. We're going to move to a slightly different lung cancer system with a different mutation and some different drugs, but really the same idea. So again, we're going to set up the same experiment and we're going to start measuring some different games and see if we see more than what we saw before. And I think what's really fascinating is a few things. One is that before I showed you with and without drug, but what we're finding is that the dose of the drug actually also matters immensely. So low doses of drug can give you low versus high doses of drug can actually give you qualitatively different dynamics. So if you look at the lapatinib, which is those red ones, what we're seeing is we're moving through 3 fundamentally different quadrants and steady state scenarios based on the amount of that selective pressure. And what's exciting to me about this plot is it suggests a few things, some scary things. So some of the work that Ishan's doing, a PhD student with us, is thinking about pharmacokinetics and dynamics. Because when you take a pill, the steady state level in your drug or in your blood goes up and then down. And you take the next pill, it goes up and then down. So I'm wondering if we're actually moving between evolutionary regimes during the course of patient's therapy. And if so, what is that? How does that change the way we think about evolution of resistance? And further, if you look at the black trace for osimertinib, this gives us an opportunity where we really have what we'd love to have for adaptive therapies, which is this idea that we can play with the proportions of sensitive versus resistant cells in a meaningful way. And if we could, we take the drug off and we go to the upper left-hand corner and we promote the ancestral phenotype, we turn the drug on and we go to the lower right-hand corner and we promote the resistant phenotype. Can we go back and forth, take the drug on and off, and do meaningful perturbations? I think that it suggests that some of those fundamental hypotheses that were put, actually those weren't hypotheses, they were postulates that were put forth that said, hey, we're going to try adaptive therapy. We're going to postulate that this fundamental principle exists, but I think this is evidence that it does, and that this is really feasible. And then further, I think there's this really cool phenomenon that competitive exclusion really does exist, and that's the idea that in the absence of therapy, what you really have is this opportunity for so this is an exciting result. It's just a preprint. I'm going to show you one more cartoon and then stop. But what we found, and we've found in now many situations that we've measured, is on the right. So there's two pictures you're looking at there: no competitive exclusion and positive competitive exclusion. In the one on the right, what you're seeing is that the ancestor is more fit at every proportion, except when the resistant is almost zero, but very, very close in small numbers. So what most people measure is those solid dots, the solid red and the solid blue. And what we see is that the ancestor is more fit in monoculture than the resistant. And if you have something that's more fit than another thing and you mix them together, you'd expect the ancestor to just win out. And if you just do monoculture experiments, that's all you'll see. And so on the left is a monoculture experiment, and you see that the parental is far more fit than the resistant. So one would predict from standard population genetics, you mix them together, you just outcompete with the parental. But what we're finding is through co-culture experiments that when there is an ecology and is a set of interactions, the resistant fitness is actually boosted by the presence of the sensitive and ancestor. And if there's only a few of the resistant around, their fitness is actually equalized. And that allows us this scenario that kind of gets at, this paradox that exists, I think, which is how does pre-existing resistance stick around, and why is it such a common thing? And we see this in we see this in viruses, we see this in bugs, we see this in cancer. And I'm just going to show you a cartoon that suggests, and I meant to move this slide further. Oh, it's the next one anyway. And I'm going to end here. But I think that what this shows is that this ecological effect is something that was really necessary for pre-existing resistance to be able to stick around. So in this cartoon, you see on the top, if you don't have any ecology, these costly pre-existing resistant mutations will always be popping in and out of populations because mutation is random, but they should be outcompeted quickly because they're less fit. However, if you allow also for ecological effects to be at play, you're going to have some of these costly resistant mutations that have positive ecological effects that can stick around for long periods of time in populations and hang out and persist, even with this fitness detriment. And we really think that this is a solution to some of this paradox to help us understand. And so I think there's a lot of exciting potential. I'm not going to go into the science of this, but you should check out the preprint. And again, this is done by Jeff, super talented postdoc we have who's on the market. But I think it really gives us some ideas in cancer therapy and infections as well to think about, can we not just focus on killing the cells, but can we focus on modulating the ecological effects as well? And if you could, maybe you could turn down these eco effects and really cause these pre-existing mutations to be outcompeted naturally before you even begin therapy. And I think I'd like to end there because that'll give us time for discussion. I have tons more stuff I could show you about that work, but I want to just give one more cartoon idea. And I think that what we're working on now, I think I've shown you this in vitro experimental timeline, but really there's a lot more complexity we could add. To Neil's point, we've only shown what happens when you put fibroblasts in, but we're starting now to add T cells and macrophages to these in vitro experiments. And then of course, in vitro experiments are also pretty poor models. And so we're thinking about moving from short time scale in vitro models to long time scales to 3 dimensions and further eventually into animals to see if any of these ideas continue to play out, how we have to change around our model systems, what we can learn from adding the complexity. But I think starting with that simple scenario is the place to go. And I'm going to end there so we can chat. But I want to thank the group. I already highlighted the folks that did most of the work, but we have a lovely, diverse, and inclusive group here in Cleveland. filled with folks from diverse backgrounds, both educationally, professionally, and culturally. And we'd love to have you consider come joining us or whatever. So I'm going to end there and I'll stop sharing, I think, as well, so we can chat. Does that make sense?
Yeah, that'd be great.
Cool.
That was wonderful. Vaughn, do you want to take over? I think I gave Vaughn a bad link, so he was a little late. But thanks, Vaughn, for being here.
No worries. Thank you for emceeing this. Thanks, Jake, for a fabulous talk. I'm gonna since Neil's already had one crack, we'll start with Edmund first for the first question. And please raise your virtual hands or put a question in the chat, and I'll try and call on you in order. Go ahead, Edmund.
Hi, Jake. Hi, Jake.
Hi,
Great seeing you again. I saw you in 2012 at the Society for Biomathematical for Biomath in Knoxville.
Awesome.
And you've come a long way since then.
We all have. Nice to meet you again.
You and everyone else emphasize genetic heterogeneity in the tumor, which is fine. But I'm going to introduce you to the concept of behavioral heterogeneity, which in the bacteriology world is persisters. But what I've been thinking about, and Joe Alcock certainly knows where I'm coming from on this, if you recognize that the act of growing takes resources and synthesizing to proliferation is vulnerable to stress of any kind, radiation, chemotherapy, any kind. And those are the proliferating cells. But within any population, whether it be people in any population, bacteria, tumor cells, there's going to be heterogeneity in their potential response to stress. So you can proliferate and keep going. Those are the ones that are easy to kill. Just hit them with the standard stress, assuming they're all genetically identical. So we don't need any genetic variation. There's some that are going to take the strategy of let's fight the stress. Let's pump out the chemotoxin. Let's put in more free radical scavengers for the radiation effects and so forth. Okay, so that's the strategy of fight it. There's the other strategy of let's just go dormant. Let's just hunker down, stop our metabolism. And then the other strategy, and if somebody can come up with more, I'd appreciate it, is to leave. It's too stressful, let's just leave. Now, for tumors, that means that's invasion, that's metastasis. So I can see this is probably not this behavioral heterogeneity. Okay, so that's within a population, but in an individual, whether it's an individual bacterium, tumor cell, person, there's going to be a range of genes that are already expressed, proteins there. Some are for predispose that individual cell to proliferation, but there's a little bit of, hey, maybe we should be ready to leave genes or proteins available. Some might have the dormancy tendency. So within a population, there's a variety of behavioral choices, bet hedging. And within an individual, there's a degree of bet hedging. And so what I'm thinking in some of the models you've done, these in vitro models, there's not a lot of environmental variability. You know, in a plate of, there's some, but in a plate of antibiotics, there's not a lot of variability. But if you think inside a tumor, now you've got distant, are you on the edge of the tumor? Are you in the middle of the tumor? How close are you to blood vessels and nutrients? And so there's a whole range of variability or an infected site. And so where the individual tumor cell finds itself should dictate a tendency on its behavior. A cell on the outside of the tumor seeking better conditions, gee, maybe I should crawl away from here and see if things are better on the outside. That's not a good strategy if you're in the middle of the tumor. And if you're if you're in the middle of a tumor right beside a blood vessel, hey, conditions are pretty good. Let's go for proliferation. So my point is, it's the same problem though with the genetic variability. Genetically, the sensitive ones are easy to kill. It's the resistant ones that aren't. And in this behavioral point of view, the proliferative ones.
Can we wrap it up? I want to move on to some other stuff too.
Okay,
Thanks.
real quick. The proliferative ones are easy to kill, but the other non-proliferative ones are tough to kill. So, but it'd be interesting to model from this other aspect. But I think the answer is going to be about the same. Easy to kill the sensitive ones, hard to kill the others.
Yeah, totally agree. I dropped some papers that can address some of those things in the chat if people are interested.
Oh, good.
I don't want to go. I would love to. To your point, it's extremely well taken. The most recent one's a preprint from Ishan, who's the student I've mentioned a few times, that really thinks about how to move some of these things into spatial models. And I think, mathematically, it's relatively straightforward to move some of these things into spatial context with vessels. And the preprint I just dropped is more about microenvironmental gradients writ large. There's vessels modeled there. And the one there is genetic variability, but I'll suggest it's relatively straightforward to change that from genetic to phenotypic variability without any changes. So certainly that's the direction we want to move. And your point's incredibly well taken. You do not, and if you notice, I actually didn't mention genetic differences in the the first paper. We're talking about a population of stuff that evolved to be different from a population of stuff that wasn't. We did sequence stuff because reviewers like that for those journals, but that wasn't the point of the paper. I don't even care how they're genetically different. I care how they're behaviorally or phenotypically different and their competition dynamics. So your points are super well taken, and I think it's incredibly important, and I'm psyched to keep working on it. Thank you. You're muted. Yeah.
Yep. Neil, would you like to unmute and ask a question?
Sure. So at the very beginning, I just noted that I really don't like that term that you used about resistant bacteria with the word prefix.
Super. I know you hate superbugs, Neil. We've had
it's not his term, in fairness.
It's not my term, and I only use it on one slide, and I apologize to you on that slide. So
well, it's not about me. The point is that you spent a very stimulating seminar building the case of why that's a stupid term.
I'm glad that we agree.
So that my point is, people in the evolutionary medicine arena should be the individuals most keen to push back against this overly simplistic kind, and it's not just this term, but very simplistic kinds of schemes. I think it's worth the extra effort to find a way to help other people who aren't focused on evolution to understand why it matters and to take it into account and not settle for the easy way out.
I'm going to drop Neil's paper on why we shouldn't say superbugs in the chat.
By the way, the editor of the newspaper in question, despite the fact that we did not use the word in the title, put it back in the title. And the whole point of that title was to not use the term. That's what you do with editors who think they have the right to entitle the paper. Anyway, thank you.
No problem. And I agree. It's an inappropriate term, but it is. All terms are wrong, but some are useful. And I would argue that yes, yeah, indeed. Thanks, Neil.
Thank you, Neil. James?
Can you hear me?
Yes. Hi. Oh, we could.
Oh,
there we are.
Hey, sorry, I had a quick flip there. I've had much pushback from people saying the PK/PD is the only thing that matters in these cases. For example, when I've been doing some work on adjuvants and antibiotics, we've talked about changing the rate of the adjuvant to the drug. And the biggest pushback I've had is, well, the reason we have that ratio is because that's the most effective PK/PD. Have you had that criticism?
So we're actually just getting. So Ishan, I might ask you to say something if you're actually physically present. Do you want to turn your camera on if you are, and if you're not listening, don't do it. So we're just getting into the PK/PD world, and I think again, Ishan King, who's just there, right? Wave Ishan so they can see which one you are, is a MD PhD student doing some work who really has started to move us toward this, and I really do feel like your point's well taken, which is that the differential dosing vastly changes a lot of stuff. So Ishan's preprint that I put in the did you maybe put your other one as well there, Ishan, if you will, really shows that differential drug dosing can change the biasing of fitness. So it can literally change the trade-offs between which type or mutant is more and less fit. And then further, I think we just showed too that it also changes the ecological dynamics. So I think it's massively important what's happening. And previous to the most recent work, I've been stuck with yes drug, no drug. And I do feel like we've been missing it, but it's been really hard to incorporate those evolutionary and ecological dynamics with differential dosing into models because they really haven't been measured until I'd like to suggest now. And so I think, and I'm sure other people have done it as well, and I've dismissed it. But our own labs work, we're feeling like now that we're directly empirically measuring some of these differential ecological and evolutionary dynamics under those differential doses, we can really get a handle on moving toward a PK/PD, highly resolved PK/PD type of model of these evolutionary dynamics. Does that answer the question? And Ishan, do you want to add anything to that?
Sure. So I guess I didn't fully understand your question. Are you talking about like clinical pharmacokinetic modeling to like optimize drug dosing and like clinical trials, like that sort of thing?
From direct experience, when I was on the job market, one of the talks I gave was just people who helped develop Pip/Tazo as a clinical drug. And they took massive exception to me saying this might not be the best ratio, evolutionary speaking. If we use a different ratio, we end up with a place where we can reuse the drug again later. And their entire point was the reason we use that ratio because it had the best PK/PD value to actually treat the patient at that time. And I guess I'm asked like.
Yeah. No, I think that's a disconnect between the clinicians and people who are really in translational medicine, not fully appreciating the impact of tumor and in this case infectious populations evolution. And so I think that we're just sort of starting to. It's been there's been some research over the past couple decades on incorporating PK/PD modeling and evolution of populations, but I think that needs to move into more clinically relevant models in terms of our experiments to really understand the next steps and how we can then go back to optimizing our dosage regimens, because a clinical pharmacologist is trying to, like you said, optimize for a patient and try to optimize in treating whatever tumor or infectious disease the patient has. But they don't really incorporate evolution into this modeling. So I think it's just a disconnect a little bit. Yeah, but good question.
And I'd throw out there in your defense as well, when it comes to combinations like Pip/Tazo, it's even, forget it. Now you have to do like a checkerboard assay.
Well,
and it just gets really, really complicated, and it's a lot. And I think, but I think we do have to move toward that. Once you realize that the eco and evolutionary dynamics are affected by those differences, you have to. It's going to be hard.
Well, I was trying to point out to the person as well that the drug availability, even through obviously a millimeter away from that hill to 10 millimeters away, they have different diffusion rates. So it doesn't matter if I think it's important from an evolutionary point of view. It happens in situ. Like it doesn't matter what I do ratio wise. It happens.
Yeah, absolutely correct. So click on the preprint I dropped in the chat, and we'd love your feedback on that.
I hate to bring this really wonderful conversation about how we need more sort of really careful evolutionary models and tests of combination therapy of drugs with differential diffusion rates. This is awesome, but we are actually past the hour. I want to thank everybody for tuning in. I want to thank again our sponsors and supporters for this program. Thanks to Johnny for leading the charge. Johnny, any last words to send us off into the holidays? I think this is the end, last Club EvMed for 2023, and it's been an awesome year. Johnny, anything else?
No, thank you. Thanks so much, Vaughn and Jake. That was really great. And I had mentioned earlier, stay posted. I'll send updates. Charlie and I are going to be working on the next semester's schedule, and we've got a lot of our committee members' ideas that we're kind of floating around. So we'll be putting that together soon. Thanks so much, everybody.
Thanks so much for having us. It was a lot of fun.
Have a happy New Year. See you in 2024.